Welcome to Học Tiếng Anh với Joseph, where we build vocabulary and practice pronunciation through reading. Continuing with The Cat in the Box, a history of science in 100 experiment. Today we will read story number 97. Hôm nay chúng ta sẽ đọc truyện số 97, Mapping the Human Genome. Just 70 years ago, the span of a biblical lifetime, the chemical composition of genes was still a matter of debate. Less than 60 years later, in 2001, the composition of every gene in the human body, the human genome, had been mapped. Armed with this information, it would be possible in principle, given the technology, to make a human being out of basic chemicals. More practically, it opens the way to cure diseases such as cystic fibrosis by genetic engineering, repairing faulty genes. DNA, the stuff genes are made of, is composed of a string of bases known as by the letters A, C, G, and T. These can be thought of as the letters in a four-letter alphabet. Instructions for the construction and operation of the body at a cellular level are coded into genes in the form of three-letter words such as AGT, GAT, AAC, and so on. These are known as triplets. A string of these words contains information, just as a string of words in our alphabet contains information. Any information can be written in this way if the string of letters is long enough. After all, any information can be written in the English language can also be written in the binary code of computers, which contains only two letters, 0 and 1, on or off. That is how we are writing this book, and maybe how you are reading it if you are using an e-reader. The string of letters in DNA is long enough to contain the information required for human life. The human genome contains roughly 3 billion letters, equivalent to about a gigabyte of computer memory. In a sense, this is surprisingly small, which is what made the project to map the human genome possible, but it applies to each individual cell. Thanks to gene mapping, we now know that there are between 20,000 and 25,000 separate human genes arranged in 22 paired chromosomes, plus the X and Y chromosomes that determine sex. Mapping a gene or a genome is technically known as sequencing, finding the sequence of letters along a strand of DNA. The experimenters also need to find out where one gene ends and another begins along a strand of DNA that makes up a chromosome. To do all this, they use enzymes which act as chemical scissors to cut DNA into small pieces at precise locations that depend on finding an exact string of letters, like finding a particular word in a book. These fragments are then separately separated by a technique called gel electrophoresis. The bits of DNA are put at one end of a tube filled with sticky gel, and an electric current is used to drag them through the gel. Small fragments encounter less drag, so they move faster than long fragments, and the bits get sorted by size. Then the fragments are analyzed chemically to find the sequence of bases along them. This involves amplifying the fragments by making multiple copies of them using the same techniques that the body used to copy DNA, so the chemists have a brew containing many identical copies of a stretch of DNA to work with. The idea of sequencing the whole human genome was proposed by an American researcher, Robert Sinsheimer, in 1985. This led to the establishment of a project under the auspices of the U.S. National Institute of Health, NIH, in the early 1990s. But one of the NIH researchers, Craig Venter, left to set up his own genome project using a slightly different technique. This was a commercial venture under the name Celera. The result was a bitter rivalry and a race to map the human genome, which culminated in 2001 in a brief truce when the two groups, each involved thousands of researchers, published their maps. By agreement, Venter's team published in the journal Science, and the NIH team, by now known as the International Human Genome Sequencing Consortium, in the journal Nature, both in the second week of February 2001. In truth, neither map was complete. Both teams recognized that their sequences were first drafts. Each of them has sequenced something over 90% of the human genome, with gaps about 100,000 letters long. The rivalry to complete the task soon became as bitter as ever. Celera now focused on developing drugs based on the genome information, while the consortium pushed on to plug the gaps. As a result, there were several subsequent claims that the mapping was complete, and the INH in particular prefers the 2003 as landmark date, but as American commentator Laura Helmuth, who has followed the saga throughout, has pointed out, celebrating 2003 rather than 2001 as the most important date in the sequencing of the human genome is like celebrating the anniversary of the final Apollo mission rather than the first one to land on the moon. To the vocabulary part. Biblical lifetime. Biblical from Bible. Debate. Human genome. Sequencing. Electrophoresis. Encounter. Fragments. All species. Bitter rivalry. Bitter with two T nha. B-I-T-T-E-R. Bitter. Culminate. Sequencing consortium. Landmark. Thank you for your time, and I will see you in the next one. Bye for now.